It's with my distinct pleasure to introduce Dr. Ralph Brown today as a lecturer as part of the CCOVI lecture series. Dr. Brown is he's now retired professor from the School of Engineering at the University of Guelph, and he's been a CCOVI Associate Fellow since 2007. He has many educational achievements, including Bachelor's of Science and Master's of Science from the University of Guelph in Crop Science, as well as Bachelor's of Science in Engineering from the University of Guelph in Biological Engineering, as well as a PhD in Biological Engineering from the University of Guelph. A lot of his research focuses on instrumentation, remote sensing, and applied geomatics in precision viticulture. And over the years, as a graduate student in the viticulture lab here with Andy Reynolds, Ralph was a collaborative researcher with a number of projects, and I had the pleasure of working with him and some of his students in the vineyard as well. And as a viticulturist, it's always nice to have engineers working around you, and makes you a bit jealous though. It's almost like Jack Nicholson in the first Batman movie. Where does he get those wonderful toys? So it's my distinct pleasure to introduce Dr. Brown today, and his talk is titled New Tools to Fine-Tune Quality Harvest, Spectroscopy Applications in Viticulture.
Thanks very much, Jim. I'm glad you didn't tell any of the stories from our excursions and all of the expeditions out in the vineyards. Anyway, yeah, as Jim said, the talk that I'm going to be presenting here today is on basically techniques of spectroscopy applications in viticulture. And I am interested in viticulture, and I'm interested in instrumentation, so this is kind of a nice marriage of the two. And the introductory slide here shows a couple of different applications. Here on your right, we have a near-infrared spectrometer, the Brimrose system, and Larry Gibson is holding some grapes up there. We're taking some measurements, and that's one application of spectroscopy. And here's another application here. We have Dave Lederhoff in the background. Actually came to Brock after he was working with me on his project, and he did a project here doing his master's. And what he's doing is another spectroscopic application where he's using a portable spectrometer to measure the reflectance of leaves in the vineyard. Anyway, the little bit of background first, I guess. The question, why spectroscopy? And what's the interest in terms of viticulture? And really the interest is that as viticulturalists, we want to produce the best quality of fruit so that we can get really outstanding wine. And to do that, we need to know when the optimum time to pick our fruit is. And of course, we can measure different qualities of the fruit, such as the solids content in sugar bricks. We can measure pH. We can measure other variables in the lab. But it would be very nice to be able to do it quickly so that we're not collecting samples and having all of that extra analysis and the extra time. So the idea here is to use light, which interacts with matter, and then use the characteristics of that light, which may be absorbance or reflectance or transmittance. Because the way that light interacts with matter is reflected by the chemical composition of the material it's interacting with. And the way that happens is that the atoms and the molecules that make up these different compounds have different energy levels based on things like molecular vibration, rotation and spin energy of the electrons, and that sort of thing. And all of these are specific to what we call quantum energy levels of light. And therefore, certain wavelengths, certain energy levels of light will cause certain parts of the molecule or the atom to react or not. And the idea then is that if we can shine light or transmit light through either wine or must or perhaps directly on the fruit, then the characteristics of the light are changed, and we can look at the characteristics of the light, and using a concept called Chemmetrics, what we can do is we can decode from the light interaction what the chemical makeup of that is. And so that, in a nutshell, is what we're interested in. In terms of fruit quality, what we're interested in would be, for example, the total solids or the sugar concentration in degrees Brix, titratable acidity, pH, phenolic composition, anthocyanins in the case of red grapes. So it would be very nice to be able to have, like they do in Star Trek, where they go out on an unknown planet and they shine a little handheld sensor out and it tells them everything about whatever's going on that planet. Well, it would be nice if we could do that on Earth. But we have to know a little bit about the electromagnetic spectrum in order to do that. And of course, this is the electromagnetic spectrum. In the middle part here, we have what we call light. We have gamma rays on the high energy end. We have radio waves and Wi-Fi and stuff up at the other end. In the middle, this narrow little batch here is the visible light range, and that's what we're restricted to if we just use our own eyes. But of course, the electromagnetic spectrum is much broader than that, and there's a lot more information. It's just that we can't necessarily experience it directly. Now, a lot of this information in terms of chemometrics takes place up here in the mid-infrared part of the spectrum. We can't see that energy, but instruments can be made to measure it. In terms of how some of these different compounds react, of course, chlorophyll, we're all pretty familiar with that molecule, complicated molecule. This is essentially the absorbance characteristic of chlorophyll B and chlorophyll A here in green. And of course, we know green plants look green to our eyes in a visible spectrum, and that's because on the absorbance scale, you'll notice here in the middle part of the spectrum, which is green, chlorophyll doesn't absorb very much light. So the green light gets reflected back, so we see leaves as being green. At the same time, you see these absorbance peaks, and in this case, it's in the blue part of the spectrum for chlorophyll A and B, and also there's another absorbance peak in the red part of the spectrum. That's where chlorophyll gets its energy from, not from the green part of the spectrum. Anthocyanins, another class of compounds that are really important, particularly in red grapes, occur in the skins of the red grapes. They will absorb green light and reflect blue and red. So something that has high anthocyanin content is going to be somewhere between blue, red, or purple in color. If we look at particular smaller molecules such as water, the OH bonds in water will preferentially absorb light in these ranges, 760 up to 1940 nanometers. Actually, the principal or the fundamental absorbance peak is higher in the mid-infrared, but we get all of these overtones which bounce back into the shorter wavelengths. So the bottom line is we can see some of these interactions, and if we can interpret them, then we know what it is that we're looking at in terms of its composition. So that brings me to my first application, and this is this instrument called the Luminaire or Le Vigneron. It's an instrument that the Brimrose Corporation has developed and it's commercially available. This particular one, Andrew Peller Limited had this one, and Michelle Stranges at Greenseeker worked with us on this project, so we had access to it. The idea there is we have this infrared spectrometer, which is this device. We have a grape, which we're holding up against it, and what it's doing is emitting infrared radiation and then taking the reflected radiation back again, seeing what the difference is, and then you can't see it very well, but it plots it on here. So that's the characteristic for that particular grape. And if you want to use this instrument, what you have to do is take a whole bunch of grapes, one at a time, hold them up against it, and take a gazillion of these things, and then you interpret the quality characteristics of those grapes. Kind of nifty, but it's time consuming, obviously. Each grape gives you one reading, and if you want to have a representative scan of, say, even one row or one block in your vineyard, you have to take an awful lot of single grape readings to get a representative sample. So that's a little bit of a problem. You can take it out into the vineyard. It's loosely portable. It actually is fairly sizable and heavy, but you can lug it around if you want, and you can take individual readings on each grape if you want to. One of the problems that we experienced was that the season-to-season calibration wasn't particularly robust. So if we did a whole bunch of samples, say, this year, and we developed a really good calibration which would convert our spectral reflectance into, say, Brix or pH, If we went out then next year and started shooting more berries using the same calibration, it wasn't quite so good. We would have to recalibrate. So that's a bit of a problem. The main problems with this system is the requirement for contact with a single berry. You have to take one berry at a time, roll it up, and touch the waveguide with it. And it's relatively expensive and time-consuming. So for practical sensing and practical evaluation of fruit in the vineyard, it's somewhat limited. There is a similar instrument which is under development. This is coming from a study in Chile, and it's a similar type of situation. There's a little optical probe which you hold up against the berry, and in this particular case, it uses a less expensive visible and near-infrared spectrometer. The Brimrose uses mid-infrared and farther near-infrared, which requires special optics and special light source and so forth. If we restrict ourselves to the visible and NIR part of the spectrum, then we can get much less expensive instrumentation. In their studies, in the research publications at least that have come out of this study, they're getting pretty good predictions of Brix, pH, and anthocyanins for red grapes in Chile. So the R-squared values, which are the regressed values, and basically 1 is ideal. So an R-squared close to 1 is good. 0.85 in most cases, above 0.9. So they were getting pretty good performance from this little probe, which they could take out and touch individual berries. But again, it's an individual berry. So it requires contact, it's time-consuming, you need lots of measurements. Nifty for research, but in practice, it can be a problem. So that brings me to what our interests were. I worked, as Jim said, I've worked here with people in Covey and Brock for a number of years now, Andy Reynolds and his group. What we're interested in is looking at free air visible near-infrared spectroscopy. And the question, why free air? Well, what I mean by free air is that we actually have some air between the target that we're trying to measure and the probe. So we don't have to go right up and touch an individual berry, but we can point it in the general direction, and then we can get a reading. So much more, less labor intensive, easier to use, and so forth. We also get a larger field of view if we're free air. So for the distance from the target, we can see more target. But one of the real prime movers was we're looking for something that's compatible with putting on a harvester. Ideally, what we would like to see is a probe that we could mount right on a grape harvester so that as the fruit is coming up off of the belts and into the bins, we can measure it on the fly so the winemaker knows before that batch arrives at the winery what he's going to get. And maybe stops or moves to a different part of the field or something like that. But that would be great if we could do that. The other aspect is we wanted to work with the visible and near-infrared part of the spectrum. It's not as robust as working in the longer wavelengths, but it's inexpensive. We're talking about an instrument that's a few $100s rather than tens of thousands of dollars. And it also uses standard optics, standard light, or standard glass optics rather than more expensive sapphire optics and so forth. And it also gives us information from the visible spectrum, which in the case of grapes is important. You know that as the red grapes become closer to maturity for harvesting, you get more intense color. You can see that with your eyes. You might want to measure it as well. This is just a quick slide to show. This is the actual prototype model of a yield monitor that we tested with Lakeview Vineyard a couple of years ago. This is what it would look like in an exploded view. It mounts right inside, in this case, a Gregor harvester. And the idea is that it would be really nice to be able to integrate a sensor back inside here, shining down onto the table of the yield monitor, so we can measure fruit quality on the fly. That's what we would like. One of the first grad students that worked with us on this was Wade Milton, and in 2008 he did a project. And he started out, this little box here is one of these inexpensive USB spectrometers. A few $100s now. A little laptop here, so basically it controls the spectrometer and interprets the readings. And then Wade made a fairly unsophisticated test stand here where he's got 3 quartz halogen bulbs. This is the fiber optic coming out of the top that basically takes the reflected illumination back to the spectrometer and then processes it. And then down here is a little petri dish, a sample container. Now, these are table grapes, unfortunately, so this was done in January, so we didn't have any access to wine grapes when that picture was taken, but that was the intent. So we paired up with J.L. Grew and crew at Stratus Vineyards for this particular study, so Kevin Bailey and more recently Richard Blackadder being the viticulturalist there. And the idea was that we would get into the vineyard and we would sample red grapes from early post-veraison up until they came off, which may be when the snow is flying, who knows. Anyway, we went with Cab Sauvignon, Cab Franc, and Syrah blocks, so the 3 longer harvest red varieties, and we sampled them from early post-veraison until they were harvested. The idea was we wanted to collect a composite sample, so rather than going out in the vineyard and shooting individual berries all over the place, the intent was we would take a sample, bring the sample back to the lab, and then take the measurements on that composite sample, and that would give us a quick reading, which would be representative of that row and hence that block. So we dealt with 200 very composite samples, and the way we did this is we would go up one side and down the other side of a row, and we would sample the fruit clusters, making sure that we sampled the front and the back, the top and the bottom, and the shoulders of the bunches, and we did it by touch, not by looking, because we didn't want to bias the results. And we alternated between the upper and lower wires because this was a Scott Henry system we were looking at. And then we got this composite sample. We used this inexpensive spectrometer to give us the characteristic in the visible and near infrared, and we would scan the entire composite sample at once. So it was rapid, once you had the sample, relatively portable, and more representative of the vineyard block, because now, instead of one individual grape, we got 200 grapes which have been selected to represent at least that row. And then we brought the samples back and we did chemical analysis in Andy's lab here at Brock. This approach of chemometrics that I mentioned, basically it's a statistical approach where you take the characteristics of one data set, and from that you mine information to allow you to transform that into a different realm, a different set of variables. So in our case, we're looking at light reflectance, and we want to, from that, ascertain things such as sugar content, pH, titratable acidity, and so forth. In order to do that, we and many other researchers use a software package called the Unscrambler from Camo Software. And it's basically a very sophisticated statistical package. It allows us to do principal component analysis and then do models based on partial least squares and other types of techniques to take the information and draw out the variables we're interested in. So just to illustrate what we're getting here, this is a principal component analysis plot. This is the first principal component in this axis. This is the second one here. And this is our data cloud that we gather. And so what this program is doing is it's fitting these principal components to the data that you gather so that you can represent it much more compactly. And then down here, this shows the variability of the data, and this is 1, 2, 3, 4, and so forth, the number of principal components that are fitted. So the more components we use, the better explanation of the data that we get. And then we also can do some prediction based on this, so we can go backwards. And now this shows us partial least squares regression estimates of what the parameters were based on the data that we collected. Don't need to explain this in much detail. It's a tool that we use. It's quite sophisticated. I didn't write the software myself. I'm not even sure I can use it properly, but my grad students certainly could. Anyway, here's the outcome of that first year of study with Wade. He used the Brimrose side by side with his visible near-infrared spectrometer. And here are the Brimrose readings. So the variables that we're interested in were the sugar content in bricks. So the Brimrose gave us an R-squared value of 0.7. So still a fair ways away from 1, but pretty good. For pH, we got a reasonable 0.595 or 0.6. Titratable acidity about 0.6, the same. Phenolics, in terms of gallic acid equivalence, R squared only of 0.434. So not particularly good. We wouldn't want to hang our hat on that. In comparison, the lower cost Viz NIR system gave us comparable R squared values for sugar, 0.785, actually an improvement. Much higher for pH and for titratable acidity as well. Closer to 1, better predictive ability. And 0.48 for the phenolics. The third column that I've shown here is basically the same data, treated slightly differently because here we're using partial least squares with a genetic algorithm to tune the components that we're going to include. And when Wayne did that, he was able to get, again, fairly significantly improved results down here in titratable acidity, up to 0.85. Phenolics, not too bad, approaching 0.5. Now these other figures in red here are what we call the root mean squared error of prediction. So that basically tells you if you estimate one of these variables using the system, how accurate are you going to be in terms of the units of the system you're working within. So this says, for example, that we can measure to within, well, slightly less than 1 degree Brix. So if we say it's 20, it could be from 19 to 21. So not particularly good. A little better here, 0.66. In terms of pH units, 0.05, 0.06, so reasonably good for predicting. Anyway, the outcome of that year, the 2008 experiments, were that we had some indication that this system was going to work all right. Now, you have to remember, we made quite a few changes here because now we're talking about a free air system where we've actually got 20 or 30 centimeters distance between our sensor and the fruit that we're measuring. And we're using this inexpensive spectrometer compared to a much more expensive system. And it's working pretty good. So Wade finished up, and along came Mike Feddock, another master's student, and he decided to take it a little further. So he took Wade's basic setup and he improved on the sample collection. He built basically using ABS sewer pipe from Canadian Tire. This little black sample holder here are the 3 bulbs to give us illumination. In the middle we've got the fiber optic cable. Here's a length of tube which basically just holds the grapes. You put all these 3 pieces together. This is the base. And now you've excluded the ambient light, which could create a problem. It's non-reflective, so we don't get specular reflection inside, messing up our readings. And one of the big improvements was that we could get repeat measurements by simply rotating this or shaking it slightly. Because you have to remember, we're looking at a particular area, which is a composite sample of grapes. Well, that only gives us representative information from that area. So if we mix them up a little bit, we get another reading, we get more information. And this makes it a lot easier. Just to illustrate what the spectrometer gives us, you can see from these curves, it's not very informative at all. Essentially, this is the spectral reflectance characteristic for composite samples averaged for week 1. And the only reason I present this is to see the sort of change that happens over the season. Now, all of these things are basically normalized, and if I stack them all on top of each other, as they should be for comparison, you wouldn't be able to see anything. So these are all stepped down to separate the curves. But you can see essentially where the changes are occurring, mostly out here in this part of the spectrum. So here's red in here, this is getting into the near infrared here. So this region is where most of the change takes place over the season. So the slope changes, you get some inflection points that occur at different times in the season. And there are other changes that are happening down here in the blue, violet, and ultraviolet part of the spectrum as well. They don't stand out quite as much in this plot, but there's information there as well. Well, Mike's study, 2009 and 2010, he did a similar kind of chemometric analysis using the Unscrambler software. And these are the sort of plots that he came up with. So this is Brix. And this is, the yellow O means leave one out. And basically the way that this works is we take the whole data set, we take one sample out, we take all of the others, create a model with that, and then we bring the other one in, predict what it should be, and see where it fits. And we do that for every individual within the sample, and we compare it to the calibrations using all the rest of the sample data. And this is what we got for bricks. So this is the prediction. This is a 45-degree line. If we had a perfect prediction, every point would fall on that. And this is looking pretty good. So that's bricks. Here's pH. And this is, again, a leave one out analysis. And again, you can see if you put a 45-degree line up through here, we have a fairly tight cluster. So we get pretty good representation of the data in terms of those variables, at least. So Mike's results, based on his improved version of the apparatus that he was using, these are basically the lab analysis values over the season. So as you would expect, a really wide range of bricks, because we started sampling early in September, just post-horizon, and we sampled right up until the end of November. pH, that's the typical ranges of pH, TA, phenols, and anthocyanins. And here are our predictive terms. So 0.84 for R squared for bricks, so again, pretty good. pH wasn't quite so good in this year, about 0.6. Titratable acid in terms of tartaric acid equivalents, again, about 0.6 and so forth. In 2010, again, showing the range, we had a bit broader range in terms of bricks. And here are our squared values. So 0.89 for bricks, 0.81 for pH, and so forth. We were a little disappointed in the anthocyanins in both years. It was better in 2009. We had a reasonably good prediction, but basically our standard error of prediction is still about 75 units in terms of Melvin O3 glucoside. At the same time, in 2010, we had a much poorer R squared value, less than 0.2, and our MSEP was comparatively higher. We would have expected to be able to do a lot better on anthocyanins, and we're still not sure why that's the case. We suspect that a lot of it has to do with our extraction technique in the lab and the way that we're quantifying the Melvin as well. But one of the big questions that we had from this whole project was, how transferable is this calibration from year to year? How good is this PLS model going to be for predicting next year or the year after? So the first question we have to ask ourselves is this spectral response consistent from year to year? And is it consistent between similar varieties? So if we were just to say throw some Merlot into the mix, would we get the same kind of predictions from Merlot as we get from the Cabernets or from Syrah? The other big question is, do we have to recalibrate this thing every year? Because if we do, it's of limited use to us. If we have to, take fruit samples most of the season, do the lab analysis before we can calibrate, then we're almost finished by the time we've got a robust calibration, and it's not much good for us. So can we build on each year's previous data sets? And that's a good question. So I mentioned we're using this principal component analysis to characterize the data that we're collecting. Well, this is the first principal component from 2009 and 2010 for our spectral response. You can see that the same general shape holds for both years. The actual values of these coefficients are not important. The magnitude is higher in this case as we go down. So this is a larger magnitude, but it's going in the negative direction because of the way the principal components are fitted. But what this tells us is that the first principal component is heavily weighted at this longer wavelength end of the spectrum, so the red, the near infrared. Here's another one. This is the third principal component, and again, it looks qualitatively similar. You see that in 2009 and 2010, we get this characteristic dip that occurs around 720 nanometers. So that gives us some indication that, yes, there is a similar response that's occurring in these two different years. So that's gratifying. The other thing that Mike did was he took his 2009 model, which was prepared from the samples collected in 2009, And he used that to predict the 2010 values. So based on the spectrometer reflectance of the 2010 grapes in a model derived in 2009, this is the prediction cloud that he got. Now, we would have really liked it to be right on this 45-degree line, because that would have indicated a one-to-one correspondence, which would say we don't have to recalibrate anymore in 2010 because the 2009 data is doing it for us. Well, we get a fairly tight cloud, but you can see that it's skewed off a little bit. There's an intercept and slope difference here. And the three different colors are the three different red varieties that we were working with. So we do have some transformation of the calibration from the previous years, but it's not perfect. Same thing for pH. This is again the pH prediction. 2010 spectrometer readings using the 2009 model. And again, this would have been a perfect fit along this line, but you can see that it's markedly different. So that's the bad news. We don't have enough data to give us a robust model. Now, however, having said that, if we go back and we look at these things, if we collect more years of data and we add it into this data set, then one would expect that we would get a more and more robust model that's more transferable. So it does indicate that there is some consistency from year to year, but we have to keep building up this model in order to give us accurate predictions. So what's the future of this? Well, some of you may have seen Bob Wampel's talk. He was the fellow at Fresno that developed or worked with Brimrose to develop the Luminaire system. So there are systems out there that people are using, and you may have heard about them, you may even have used some of them. So what's the future? Well, currently, with pretty much any of these systems, we're getting good results for Brix, pH, and titratable acidity. Well, that's not exactly earth-shattering, because we can also measure Brix pretty quickly using an optical refractometer. So, we have instrumental measurements there already. pH, same thing. So we're moving towards a multi-function, multi-purpose instrument that can be used to give us a number of different variables simultaneously. Some of the drawbacks, well, it's relatively expensive at the moment. This need for calibration between years and the time-consuming aspect. You're going to have to go out, in some cases, individual berries, measure a whole bunch, a gazillion individual berries before you get a representative set of variables. However, there's new technology coming along all the time, as you know. So information processing technology promises us less expensive, more robust, better instruments. And I just put this one up. This is the Planck Spectron. It operates in principle similar to what Wade and Mike did with the VIS-NIR spectrometer, rather. And it's been integrated together into a nice little handheld package, which was developed by Gilles Rabatel and a few other scientists in Montpellier, based on some work they did with an instrument they called the tremblon, which basically is French for blunderbuss, which was a big sort of a thing that they held up and pointed against the grapes and took a measurement with. Well, it evolved into this. And this became commercially available, I think, in 2010, but curiously enough, when I was preparing this talk and I tried to find out more information about it on Planck's website and anywhere else, I couldn't find any. So I don't know if this is still available commercially or not, or how well it's working, but there is potential anyway that this system may be a go. And this is basically how the instrument works. It's very portable, it's slaved to a GPS, and you can just walk out in the vineyard, and you can point and shoot at all of the clusters, and you can get representative readings, rather than lugging all of your samples back to the lab for analysis. It would be nice. Okay, the second part of the talk I want to, still looking at spectroscopic applications, but now instead of limiting myself to the berries and the fruit, I want to look at what can we learn when we apply spectroscopy to the leaves and the canopy of the grapevines. Now, all of you that are involved in viticulture know there's no substitute for scouting. You got to get out there, you got to walk in the vineyard, you got to look. You got to look for disease, you got to look for plant stress, you got to look for raccoon damage, whatever, everything. So there's no substitution for visual assessment. However, the problem with visual assessment is you're using your own eyeballs and you can only see in the visible part of the spectrum. So the question is, if we use instrumental spectroscopy, is there other information that's there in the vineyard that maybe you can't see with your own eyes, but can be useful to you? So for example, if we look in the ultraviolet or the short wavelength part of the visible spectrum or the near infrared, maybe there's information there that's useful for us. Oh, by the way, that picture that I was showing there, that's me in an Australian vineyard with what they call portable spectrometer, an ASD field spec, measuring the spectral reflectance characteristics of grape leaves in New South Wales. Now this picture, which comes from CFIA, it illustrates how, in fact, problems with vine stress show up in the foliage. And of course, you all are probably familiar with these types of images. This is leaf roll virus damage. And of course, you don't need an instrument to tell you that that's going on. All you have to do is look at it. But this sort of highlights what I'm talking about here, is that problems will show up in the canopy, which may indicate problems of balance with the vine, of stress, and so forth. These often will show up in terms of the photosynthetic apparatus. So changes in chlorophyll, changes in carotenoids, xanthophylls, some of the other pigments, chromophores that are associated with photosynthesis. And these absorb and reflect in different parts of the spectrum, and they cause changes in the leaf reflectance. So if we instrumentally can pick up on these, maybe we can learn something. This has been the subject of a lot of study. Certainly diseases such as powdery mildew, environmental damage such as ozone damage is known to cause damage to the photosynthetic apparatus. So increased reflectance in the green to orange part of the spectrum and a decrease or unresponsive red part of the spectrum. Carter did some of this work in the late 20th century. We also know that when we have things like deflox or root louse infesting original rooted vines, then that typically will show up as an increased green reflectance before we can actually see visible damage in the vineyard. Anthocyanin biosynthesis in leaves, we saw anthocyanins in the virus leaf roll problem there, but also drought, extreme temperatures will cause reduced green reflectance. So you get more purpling of the leaves and so forth. The near infrared part of the reflectance is relatively constant, even though some of these environmental stresses are occurring, with the exception of extreme water stress. And I mentioned earlier that the OH bonds preferentially show absorbance troughs in the 1940, 1450, 970 nanometer range. Well, if the water content of the leaf is really low, then you're going to start to see changes in that part of the spectrum as well. Now, a lot of these studies on vegetative apparatus in the leaves use not just the raw spectral characteristics, but they use indices that are derived from the spectral reflectance. One of the most popular of these is called the Normalized Difference Vegetation Index, or NDVI, and it's essentially looking at photosynthetically active biomass. And the way that it's calculated is by taking wavelengths in the near infrared part of the spectrum and taking wavelengths in the red part of the spectrum and creating a normalized ratio. So it's the reflectance in the infrared minus reflectance in the red divided by the reflectance in the near infrared plus the reflectance in the red gives us a value between -1 and +1. And this is widely used in remote sensing and in other types of instrumentation as well. Another ratio that we can use is the normalized green-red reflectance, or NGRR. And in this case, instead of just taking the red reflectance, we take the difference between the red and the green. And as we know, in many of the stress responses, we get an increased green reflectance and a reduced red reflectance. So if we take the two together, then we can sort of amplify that effect. So that's another ratio we can use. So the big question then is, knowing all of this, knowing the physiology, knowing the physics that's involved with it, can we do something useful? Well, we had a project that we initiated at 30 Bench, part of the Peller organization, and what we did was we went into the Riesling vineyard up on the Beamsville Bench, just west of Beamsville. We were interested in various aspects of precision viticulture, and one of the things that we wanted to do was investigate the leaf reflectance characteristics to see if that could help us do something in terms of monitoring vine and, well, vine performance for sure, and maybe fruit quality and quantity as well. So what we did was we went in and we measured the spectral reflectance of fully expanded leaves, and we would do 5 leaves per vine, and we would do that monthly over the course of the season, and we would do it for 500 different vines in this Riesling block, and then we would take all of that information and look for correlations. In the same study, what we also did was we looked at soil moisture, so we used time to mean refractometry, or TDR, to measure soil moisture. We measured vine water stress or vine water status using pressure bomb. We did harvest yield and quality on all of these sentinel vines. So we had a whole stack of data that we could then investigate. Well, in my introductory slide, I showed this picture. This is Albert Brooks and Dave Lederhoff, and this is actually going out and taking these measurements. So David here is using this home-built spectrometer extension, basically. What it is a pair of tongs from Canadian Tire for a barbecue, and we have a little quartz halogen lamp on top, and here's the fiber optic probe coming out, and you can just go around and clamp it on different leaves and take measurements. And there's a hemispherical chamber here which keeps ambient light out so that you get repeatable results. So having done this many, many times for many vines over the course of the season, here's what some of the data looks like when I plot it. So here are 2 different dates. This is the normalized green-red ratio from these individual leaf reflectance measurements made on July 26th in 2007. August 24th in 2007 is down here. And these little cartoon plots basically take those point measurements and by using geostatistical techniques, we can come up with likely contours of where these different data would occur. And these are based on 500 data points, so it's not too bad. And down here at the bottom, what I have is mean soil moisture. So this is the soil moisture distribution over this block in 2007. And down here is low moisture in red up to higher moisture, greater than 16% in blue, which in 2007 there wasn't very much, because if you remember, 2007 was extremely dry year. It was a great vintage, but we had a lot of water stress. This was a non-irrigated vineyard, so what we see was a consequence of what moisture was available. And what we see is very dry areas down here and relatively moist area in here, where I've drawn this little diagonal line. If we look at the corresponding plots for the green-red ratio, particularly the later one in August, we can see this is approximately the same location. We are also getting relatively higher green-red reflectance ratio in this area where we have better water availability. Down here where we have poor water availability, we get poorer or lower readings. This is a wet spot, so we'll just ignore that. So the idea is, yes, you can make measurements of soil moisture, for example, but somehow we also have similar information encoded in the leaf reflectance. So the zonation is similar. When we look at harvest, here we have, again, that's the normalized green-red ratio plot. So here's that diagonal band where we had a higher NGRR and somewhat higher soil moisture. And here down is the harvest data that we collected. So this is the sugar content, so bricks at harvest. So here's that same diagonal area down here. Well, in this case, we go from red up to blue, giving us correspondingly higher and higher sugar contents. So we get low sugars over in here, we get relatively low sugar in here, and we get somewhat improved here, which corresponds roughly with what we were seeing from the green-red reflectance. Now you might have to, squint a little bit and use your imagination to see these relationships, but you have to remember that these transfer functions aren't direct. There's a lot of other things going on in the vineyard in addition to just these simple sort of relationships that we're looking at. But this does give us evidence, at least, that if we know something about the spectral characteristic of the canopy, whether it's in terms of the NDVI or the green-red ratio, then we can make some predictions of which part of this vineyard is going to give us low sugars or higher sugars. And that might be useful information. So the realization is that, yes, there is some kind of a relationship between plant stress, leaf reflectance, and subsequent fruit quality. But obviously it's very complicated. The correlations simply aren't enough to make easy, direct predictions. But they can give you a clue, right? Other factors, for example, the crop load that you're carrying, the weather, the pruning, trellising methods, all of these things have big effects too. However, the underlying realization here is that this leaf reflectance information does have potential to create maps of zones. So if we're interested in precision viticulture, if we're interested in treating different areas of the vineyard in a different way to capitalize on those differences, then maybe this leaf reflectance is important. For example, differential harvest. That's the typical one that people do. If you have an area of the block that has a high sugar content and you have another area that has a lower sugar content, maybe you want to take the lower sugar and make sparkling wine out of it, and take the higher sugar and try to make something better. Whoops. I don't know how the laser pointer is able to trigger that, but anyway, it seems to be able to do it. So, we use this home-built spectrometer with the barbecue tongs, and that's okay for research purposes, but if you're a grower, you don't want to have to build all this stuff yourself and test it. However, there are now instruments on the market. If you're familiar with Spectrum Technologies, who do TDRs and all sorts of other nifty tools for viticulture, they have a Field Scout CM1000 meter, which is basically a digital NDVI meter. So it's a point and shoot device. You point it at what you want to measure the NDVI of, you pull the trigger, and it gives you a reading. So instead of using our home built, you can use this, albeit more expensive, but probably more robust and certainly nicer looking. There are also these little handheld meters which you can get. This one here from OptiSciences is a chlorophyll meter, but the same technology can be used with two different optical bands to give you pretty much any kind of data that you want out of it. So this young woman is using this on maple leaves. I included it because they kind of look like grape leaves, but not so much. But that is an application. So some of these instruments are out there for handheld data collection. But then there's a whole other family of instruments out there. This one is called the Greenseeker, and this is what the business end of the Greenseeker looks like. It's essentially a two-band spectrometer. It measures in the red, it measures in the infrared, and here it is mounted on a little quad ATV, and this little fan gives you a visual indication of what it's looking at. And here's a GPS unit on here, so basically you just drive up and down the road, taking measurements all along here of the NDVI, and when you're finished, you've got a map that looks like this. So now, instead of walking through the vineyard and taking individual readings, you can drive leisurely through the vineyard or at high speed if you want. And this is the particular instrument here. Now it's looking on both sides, and you've got a happy fellow on the quad going through here. Amazing. Maybe he's done some spraying at the same time, but I doubt it. Anyway, the intent is that you come up with this NDVI map. Now you can see where he's gone up and down and up and down. And different colors indicate here's blue and green up here, and here we have red and orange down in here. So that's giving us relative differences in reflectance. So this might be a high vigor area, or it could be a low vigor area. I don't know how this one is interpreted. But that's the intent. I was in New Zealand a while ago, and a fellow named Thompson is doing some work commercially on this. Here's a couple of different blocks. This is a Syrah block here, this triangular one. This is a it's Merlot block here. And this is one of these NDVI maps that's created from the Greenseeker. Now what they did was they went in and about here, they sampled and harvested this part of this block independently from this block up here, and then they made two different batches of wine. One was able to command a premium price, and the other one was sort of a table wine type price. So they've capitalized on this, and in fact they offer this service. Spatial Solutions in New Zealand you can contract them to go out and do NDVI maps of your vineyard, and then you can use that information. So closing up, is there potential for using some of these vineyard canopy reflectance observations in precision viticulture? Well, certainly we have the Greenseeker and the Crop Circle and probably other systems that are out there right now that we could use and are being successfully used in Australia, New Zealand, parts of Europe, South America, not so much around here. Although I know Cornell, I think, has a Greenseeker setup, but I'm not sure if anybody up here has used it. There is certainly evidence, ample evidence, that NDVI variation is related to harvest quantity and also somewhat less directly to harvest quality. So if we know what our vineyard looks like in terms of a map, then we can do something about it. This may be more convenient than remote sensing, which would be another approach where we use either an aircraft or an unmanned aerial vehicle or a satellite in some cases to basically take measurements on the ground in a snapshot format. So rather than having to drive up and down, you just take one picture and get the same information. That sounds convenient, except it's basically weather dependent, so you can only do it on a nice sunny day. If it's cloudy, you can't see anything through the clouds. So if you have your quad bike and your side-looking NDVI sensor, you can go out and you can still do it. Doesn't matter if it's cloudy or not. So it may be more convenient. What we definitely need is more research on the linkages that occur between the vine balance, the spectral characteristics, vine health, and fruit quality. So there obviously is some sort of a transfer function that exists, but it's really complicated. And unless we hold the other variables constant, such as the crop load and the pruning methods and nutrition and that sort of thing, we won't make very much progress in determining what that is. But I think potentially we can do it. We can certainly do it in terms of limiting factors. So there is research needed, but we do know that essentially a well-balanced vine, a vigor under control, good fruit load produces better quality than the opposite, where we have an unbalanced vine, maybe too much crop load, and not as much quality. So that's what I wanted to present here today. A couple of different applications. The application of chemometrics and spectrometry in terms of fruit quality, and using the spectroscopy in terms of the vegetative characteristics and what we might be able to learn from that. So that's a couple of spectroscopy applications in viticulture. There are, of course, more. The one that you're probably all most familiar with is instrumentation like the Foss wine scan or something like that, which is a mid-infrared optical instrument that's designed to give you the chemical composition of wines and musts. But in that case, we're talking about a liquid sample, and we're talking about quite an expensive, well-calibrated piece of equipment, which many wineries now have. That's also a spectroscopic application, but that's more in enology and not so much in viticulture. Anyway, with that, I'll say thank you very much for your attention, and if you have any questions, I'll try my best to answer. Thanks.
Thank you very much, Ralph. It was a very interesting talk. Explain some of the new applications that you've been working on over the last 4 or 5 years or so. I'll open the floor for any questions. First off, are there any questions in the room?
I'd be surprised if Kevin didn't have any questions for us.
Are there any questions online at the moment? I guess the one thing that was interesting was, I mean, we have so much variability in Ontario in terms of, especially for raising to harvest. A lot of these applications are used in, I assume, Australia, California, where there's not as much variability probably from raising to harvest. And the one encouraging thing that I saw from your data was 2009, 2010 were completely different years in terms of, especially September through the harvest. Yeah. And it was encouraging to see some of the prediction models that you did get. How many, like in the same issue with the wine scan and so on, is that the data isn't really as good as the equipment's calibrated and information that's in the library, I guess we can say that.
Yeah.
So how many years would you think it would take? Like, do you see this being relevant in Ontario after more people? And I guess you have to use it to.
Right.
The more people use it, the better it would be, I guess, right?
I think that's an interesting question coming from you in your position, because if you're representing the growers as a group, yes, some of these technologies require a lot of calibration, a lot of sampling for validation and verification. So if it would be possible to coordinate an effort with a whole bunch of growers where you could sample all over the escarpment or all over the peninsula on a regular basis, and then everybody would pool their data, that would be good for everyone. Obviously, we had only a very small number of data points for this kind of a study because well, both Wade and Mike were limited to about 11 weeks of sampling. So, we're only talking about a few hundred data points. We really would like to have a few tens of thousands, if not hundreds of thousands, to make a really robust calibration. But I agree with what you say. It's nice to see that there seems to be some transfer, at least from 2009 to 2010, which are completely different years on a small number of data sets. Mm-hmm. I think ideally what we would be looking at would be sort of a bootstrapping approach, where we start with a data set and then we keep building on it year by year. So we have more and more people participating, they feed their data in, because they are doing lab analysis as well. So if we have the spectral reflectance and we have their lab results, assuming they're using the same methodologies, then we can keep, feeding this in and create a database, which would be useful. So, but that would take, quite a bit of effort by a number of people and somebody to coordinate it on. So.
Thank you. Are there any other questions? Do you see precision viticulture? Obviously, with technology, it starts off as it's a space age technology, and then it becomes the standard norm. And she demonstrated there's a lot of new equipment on the market that's accessible through spectral technologies or what have you. Do you see this being used more for larger applications, premium winemaking? Where do you see precision viticulture going in the future? Or both, I guess.
I think in the beginning it would be the smaller premium producers that would be the most able to capitalize on it, I think, because in the smaller production units, more responsiveness, more hands-on, and that sort of thing. When you get into the really big ones, where you have a lot of commercial operations that are doing things on a broad scale, it would be a little bit more difficult, I think, to have them get on the same page. Whereas the small sort of boutique winery that is really in there all the time, they probably, initially at least, would be the most interested in this. But I think once there's money to be made in any technology, there are people that are interested in adopting it. So, if you can demonstrate that this NDVI mapping, for example, on a large scale in New Zealand and Australia, they're using this, a much larger scale than we have. So why can't we use it? Well, there's no reason why we can't.
Yeah, that's
we just haven't done it. Yeah.
Thank you very much for your fantastic talk. Enjoy your retirement. I'm glad you came out of retirement for this lecture, and there's a token of our appreciation.
Great. Thanks very much.
Just a couple housekeeping notes here. We have a special lecture next Monday, March 26th, on good practices in Pinot Noir winemaking. Dominic Del TL, will be coming to speak on Monday in this room from 11 to 12:45 p.m. And it will be also through webcast, right?
Archived video.
An archive video then. And this is not any self-promotion or anything, but next week I'll be speaking here at the CCOVI Lecture Series. The title of my talk is Beating the Cold, Best Practices to Maximize Hardiness in Grapevines. So an overview of some of the research that I'm doing in terms of cold hardiness. So with that, thank you very much for tuning in. Thank you for all of you here for attending. And I'll see you next week. Thank you.